I'm going to try and make it a little bit of a story. And with that sort of theme here, I've entitled the talk, Reading the Landscape. And I'm going to do just that. I'm going to try and tell you some stories. Hopefully they'll be backed up with a little bit of inference, but for the most part, hopefully something that's fairly digestible. So I've put on barbed wire here. And I think barbed wire is a really nice metaphor for this thing that I'm calling reading the landscape. So what I see here is evidence of a dispersal thwarted. Here is a little bit of wool caught on a stray barb. And the concept of what I'm going to try and extend to you today is very similar to this. Can we, or how can we, use genetic information to read something about the landscape? Here we're learning, perhaps, that a fence represents resistance to a poor sheep. But we can use genetic information in similar ways to tell us stories about landscapes. So, the topics. I want to start with why and how to read the landscape. I'll go through the background related to landscape genetics and landscape genomics, which are these sort of fairly new fields. Give you some illustrated fireside stories about how landscape genetics has been used to read the landscape, and then ask at the end, can or will genomics help in this quest? So this, to me, is an archetypal landscape. I've spent a lot of time studying caribou. So for this animal, it looks like it's flat, right? There are some trees, and there's some rocks, and there's some sunshine, and there's some clouds. The animal, the caribou, is sort of like the granddaddy of all movers, can cross that landscape without any trouble. But there's a road on it. And well, you can ask the wildlife biologists, and they'll say, I saw that caribou crossing the road the other day. But maybe this is how that road actually looks to that caribou, or how it's experienced by that caribou in terms of its gene flow. And it's that kind of signal we're going to try and read from genetic information. Now, I don't want to give the impression that that road is a barrier, because there's a ladder in it. So there is dispersal, perhaps, across these features, but it's harder. So what that ladder, to me, in this cartoon signifies is resistance, is a reduction in dispersals across a feature. And it's that reduction in dispersal and in gene flow that creates the signal that we try to read. Next, let's pause for a minute and ask, why do we care? I think this is always the ultimate question for me when I'm listening to a talk. Why should I care about this? So really, there are two main reasons why we can use these kinds of landscape reading procedures. One of them is to understand how we can inhibit the movement of organisms. For example, pathogens and their hosts. If we can understand how the host or the vector experiences that landscape, if we know what causes, slows its movement, what features might affect it, we can then do something perhaps to spread zoonotic diseases. Invasive species like the zebra mussel. If we understand how features of drainages affect the movement of propagules, we can then also perhaps do something in terms of invasion. Rabies spread by this raccoon, or chronic wasting disease, a disease of ungulates spread by white-tailed deer. Again, we can ask questions about how a landscape affects the movement of the host and vector. So inhibiting movement, one of the goals of this kind of work. Also promoting movement, and this is where the conservation kinds of questions come in. You've got mountain goats or wolverines. How can we make sure they can find each other to mate? Connectivity question that can be answered using genetic information. The boreal caribou, endangered in Canada. How is the landscape affecting it? And how can we make sure they can find each other and they're not impacted by a reduction in connectivity in the landscape? Or bumblebees under climate change, can they move north as the temperature warms? Or this lemur here, which has been well studied in Madagascar. Similar questions have been asked about promoting movement. So this is the archetypal image. I have spent a lot of time looking after young kids recently, so I like really simple images here that tell simple stories. Can you imagine a sort of 2-allele system here? You've got the green allele and the red allele, and there's no road in that picture. And then the green allele is obviously fairly rare in this population. And then we build the road, and this signal might emerge potentially, if that road represents reduced gene flow. And it's reading, in much more complex terms, but reading this kind of pattern on the landscape and relating it in inferential terms to features we see that is the story of landscape genetics. So 2 levels at which we do this kind of reading. There's the population level reading, which is really the most traditional type. You can examine relationships among populations in terms of their genetic similarity and in terms of also their distance on the landscape. That could be isolation by distance analyses, or you can look and see how the resistance between those populations, in other words, the presence of that highway, does that correlate with the genetic signal? So that kind of population level reading, probably quite familiar to most population geneticists or who have dabbled in that kind of thing. Where I think landscape genetics is really going and needs to go further is to throw out about 50 years of population genetics theory and to think not about populations, but to look at spatial autocorrelation in genetic similarity among individuals. So what that means is really we see across space, are there any aggregations in the similarity of individuals? So that's really an individual level reading, and that's really what I'm going to focus on here. So just briefly to talk about, there's 2 sides to the equation. We have the landscape pattern, we have to capture that, and we've got the genetic pattern, we have to capture that. So we need both of those pieces. I want to talk briefly about the landscape pattern, very briefly, just to give you an idea of how this is done. You usually start with this thing called a resistant surface, which describes how much we hypothesize the landscape is going to affect animals. And then we do things like least cost paths to relate for the distance between P and Q. So the least cost path might be a bit tortuous here, and that might imply it's harder for genes to flow from P to Q. Another way is we throw out the whole concept of linear movement trajectories and understand the landscape as a series of connected regions. We call this approach grains of connectivity. And it kind of corresponds to this idea that I showed in this cartoon. So understanding landscapes as connected regions. And that work has come out in a series of 3 papers over the last few years. So what I want to talk now is not about capturing landscape pattern, but capturing spatial genetic pattern. And this is a paper that I published with Pedro Perez-Neto and others in Methods in Ecology and Evolution just last July. And the real goal of this is to take a bunch of animals sampled on a landscape. This is a simulated landscape. Those crosses indicate where they are, and pull out patterns in their genetic relatedness. So I don't know if you can see it very well in this room, but these gray areas are non-habitat in this model. They're areas that might slow the movement of our animals. And these patterns, following the simulation, emerge where you see clusters of genetic relatedness or genetic signal, which we then can relate back to the environment that created it. More on that in a minute. So how do we use this memgene tool that we developed to do this capturing and describing spatial genetic pattern uses this thing called Moran's eigenvector maps, which really pulls out a series of orthogonal descriptors of potential patterns in spatial autocorrelation. So you imagine you have n points, then you have n-1 possible ways that those points could covary together. And you generate these hypotheses, if you will, and then you mine those hypotheses out of the genetic distance matrix. So you've got the genetic distances, could be any kind of measure, relatedness or proportion of shared alleles, and you mine those patterns out of that genetic distance matrix, which is supported through a forward selection procedure. And then you do a final model and from the predicted values of this regression, you can pull out what we call the memgene variables, which are patterns of significant genetic structure. So looking at the relationships among individuals and their genetic relationships, these are significant spatial patterns in genetic structure. So that's really key. You've got this massive, thing called genetic variation, but we're homing in on just the spatial piece, just the part of that genetic variation that co-varies with space. And we can get an R² out of that allows us almost to estimate the degree of panmixia you have in a population. If you have 0 R² out of this regression, you effectively have the animals having no spatial genetic structure, but as that R² increases, you have less panmixia. So I'm going to talk more about this now in a series of vignettes that use real data, and that's the thing, that use other people's data. I'm that kind of nasty ecologist that doesn't act, or uses other people's genetic data but doesn't actually create it himself. And I'm going to show you all of that here. So first, before we get to that, I just want to back this up with a little very quick individual-based simulations following on Sam's individual-based simulations. Here's an individual-based simulator that I developed specifically for landscape genetics that allows us to simulate potential things in space or in the landscape that might influence movement and gene flow. So we have this sort of little radial-like structure, and the model basically, the movement of individuals and their mating is influenced by the shape of that structure. So when you run memgene on this data, after you let the animals breed and move around for 30 or 40 or 50 generations, you get this kind of pattern emerging. So clearly we've got the barrier here, and we have 3 clusters of related individuals emerging. And the R² associated with that is 13%. So that's 13% of the total amount of genetic variation we generated is correlated with space, or is explicable significantly by space. Similar, in a different landscape here where we've got a fragmented pattern, this might represent habitat fragmentation, or mountain valleys, for example, where these white areas are the lowland areas and the gray are the higher, less hospitable areas. So we let animals move and mate in this landscape for 30 generations, and we pull out a pattern corresponding with the white areas. So resistance of the landscape is influencing their movement, their mating, their gene flow, and even after all of that, we can still identify 3% of the total amount of genetic variation and use it to tell a story. Very small amount. So this procedure allows us to take very cryptic spatial patterns within populations and tell stories about it. Again, we have a river here, and we see a nice gradient that follows the river bank. So now let's talk about some real populations. Here is me pilfering somebody else's data from Dryad. These are some polar bears, and I'm not sure how many microsatellites are used here. But they're polar bears sampled all across a very large area of Nunavut. And what they've used structure, which you probably have heard of if you do population genetics, they found k equals 2, which means 2 populations in this area. And, well, we found the same thing using memgene. At least it works. But we also can pull out some other patterns, too, about what might be happening in terms of gene flow using memgene. Another system here, also in northern Canada, the Peary caribou. It's an endangered species, lives up here almost at the very top of Canada on Ellesmere Island. And these are a very mountainous area. The white areas indicate higher elevations. So again, we can pick out groups of Peary caribou that are associated with lower elevations. So topography. So this is really the first type of landscape we're reading here. Topography is having an impact on the movement and gene flow of these animals. And it's a very small amount, 4% of the total variation. Fish now, Cumberland Sound, also in northwest in Nunavut here. The Arctic char kind of go back up and forth from the ocean here to natal ponds, and they return to the ocean to breed. And what we're seeing here is a gradient that runs the length of the bay. So this implies that the location of the mouth of the river, rivers with proximate mouths flowing into the ocean are having closer genetic signal. Okay, bumblebees. One of my favorites and an area that I'm moving further into these days. This is somebody else's data again. And this is of a very small region near Sacramento, California. This is the yellow-footed or black-nosed or one of those bumblebees, I don't know. So lots, you can't really see it, but there's lots of agricultural activity going on here. And this is quite a small area for landscape genetics, but we're seeing quite a differentiation between these animals at the center of the map and the ones at the bottom here, corner. And what it could be due to the fact of that this agricultural landscape is inhospitable for queens as they move and relocate their nests, and they're not seeing a lot of gene flow happening across this area. It could be associated with these nasty pesticides we're hearing about, neonicotinoids, and the impacts those are having. We haven't really tested that hypothesis, but Shailene Jha here has tested these. So I'm just giving you pictures, but rest assured behind these pictures is some inference. Here's caribou. Nice animal that I've worked on extensively. This is also in the Northwest Territories, and here we have the Mackenzie River, which is a major drainage. So here's the impact now of major river valleys on gene flow. This is, if you run structure, k equals 1. This is one contiguous population, but yet with memgene we can pull out a nice division that is miraculously associated with the Mackenzie River. So clearly there's gene flow happening across this river, but there's a small enough resistance presented by that river that we can pull it out. 4% of the total genetic structure is correlated with it. And finally, this is the habitat fragmentation story, also woodland caribou, also in central Canada, Saskatchewan, not too far from us here. And these are woodland caribou that are very, very sensitive to habitat fragmentation and other impacts. These gray lines on the map indicate where the roads and highways are. And this, to give you a sense of scale, is about 300 kilometers across. So we're getting animals here and here separated by a highway with clearly differentiable genetic structure. And we've tested this story in many ways. And just to give you a sense that we are testing it, we can pull out the proportion of the genetic structure that is associated with various different hypotheses. So, just check where I am here in time-wise. So I just want to close off now my story. I've told you this story about how we can read the landscape. And I really haven't gone into too much detail into the genetic part of the equation, except to say that it's been there. But largely these studies have used microsatellites, so sometimes 10, 12, maybe 15 microsatellite loci. But the question of will genomics help is an interesting one. I mean, is it going to be a panacea that will allow us to read that landscape at a finer scale, to pick out more interesting, more subtle, more cryptic signals? So to do that, I did, in preparation for this, a series of simulations using the agent-based simulator or the individual-based simulator I talked about earlier, and did some of the things that Sam recommended I do and some of the things he recommended I not do. But I ran the combinations of a number of different parameters to see if having additional information in the form of information provided by genomics could actually help to the story. So I tested a series of vigility and landscape pattern treatments. So vigility implies how much does the animal move across the landscape. So we have high vigility where these black lines show dispersal trajectories. So the animal is just smoking across that barrier feature. Where in the low vigility treatment, they're not moving very much on the landscape. In fact, they're not really even interacting with the barrier feature. So this is, again, simulations of vigility. and crossing it with 3 types of genetic or genomics treatments. So starting with 10 microsatellites with 10 alleles per locus, roughly an average you might find in molecular ecology for these kinds of papers. So status quo. But what happens if we use the miracle of genomics, which, by the way, I don't really claim to understand very much, and please do correct me. What happens if we load on some of this additional information that Mason told us about in the form of SNPs, and to kind of resolve these landscape patterns better? So I assumed in 2 treatments whether we had 100 SNPs or 500 SNPs. Assuming no linkage disequilibrium. So I don't know if this is realistic or not, but I don't think it really matters here. It's more of a thought experiment. Effectively, these SNPs are completely independent in my scenario. Whether they would be independent in reality is up for debate. So run these simulations, and the question then emerges, can we read the landscape? Can we read a known landscape pattern? So we simulate the data with a known resistant structure that's slowing the dispersal and movement of animals, and can we yank it back out of the genetic data? So this green line here shows us using 10 microsatellites. So under a low vigility treatment, well, after about 15 generations, we're having about 100% success rate at reading our known signal using 10 microsatellites. And using 100 or 500 SNPs improves the story a tiny bit. So having 500 biallelic, largely independent SNPs under low vigility cases is a waste of money. However, under high vigility cases, here we have 10 microsatellite loci, and again, these simulated animals are just smoking across that barrier. So 10 microsatellite loci don't do very well. We're getting about a 20% success rate across all generations. That means about 2 out of 10 times we're reading the landscape signal correctly. If we add 100 SNPs, we don't do much better. And if we add 500 SNPs, well, we improve it quite a bit. But we're still getting about a 50% success rate. So, you might tell me, well, you can have more than 500 SNPs. But maybe you can, maybe you can't, depending on degrees of independence here. But I'm not seeing massive improvements here with but we are getting improvements when we have high vigility cases. And that's the message of this slide. This is sort of a matrix plot where the area of the squares show the degree of success of that treatment combination. So the larger the area of the rectangle, the more successful it was overall. So you can see 10 or 100 or 500 under the low vigility treatment doesn't make much difference what you do in terms of genetics or genomics. Under the high vigility treatment, having more information clearly improves things. Although overall, the width of this column indicates we're getting a much lower success rate overall. And you get a similar story when you look at this other more complex landscape that's kind of fragmented. But the message that I just can't seem to figure out is why microsatellites seem to be doing better in the low case here. And I think that's going to need a lot more work to figure that out. So just to summarize, what are the main messages here? When you've got high vigility organisms and you want to read the landscape, try neutral SNPs. Maximize information content, but please expect low power with existing methods. So this is an ongoing issue in landscape genetics. Methods really aren't very good. But it's still early days. So I think there's a lot of room for improvement, and methodological improvement is likely a major area to tackle before we start throwing the big guns and more information at it. Low vigility organisms, still a bit of an enigma. So my concluding slide, the message I want you to take home, landscape features that resist movement can leave signature patterns in neutral spatial genetic autocorrelation. So we can read, we've shown this through simulations, using real data, we can read landscape patterns out of genetic data. Robust assessments, though, of how information content, the vigility, and demography of organisms are really important now to suggest the ways forward. And I'd like to acknowledge particularly Pedro Perez Neto, who developed the memgene tool with me, and then Paul Wilson and Michelin Marceau, who have done a lot of the genetic work with me in the past. Thank you.
Well, oh, sorry. Thank you. I just like to have fun. I have a great data set for you.
Oh, bring it on.
No, I should ask you first. Have you played within populations to look at fine scale landscape features?
Looking within populations?
Yeah, just at sort of that micro scale.
Right, so that's basically what we're doing. The first slide I showed you with the polar bears, that was among populations. But everything else I show you ostensibly is within a k=1 structure population. So that's the key point here of this tool, is to get into that fine scale and toss out the whole 50 years of population genetics theory and try and read these sort of temporary, ephemeral patterns in autocorrelation and try and tell ecological stories about it.
All right, so let's thanks Paul again.